So, my name is Alice Todari. I am hosting this webinar today on biostimulant.com. The webinar is being recorded and it will be available on the biostimulant.com website very soon in the next days, and also, of course, on our social media. I invite you to use the Q&A box to submit the questions during the webinar, and we will collect them and answer at the end of the presentation of our guest. The topic of today is use of biostimulants to power up crop resilience to water stress and high temperatures, with focus on the Mediterranean hotspot. And it's my pleasure to introduce you to Professor Petronia Carrillo, today's speaker. Professor Petronia Carrillo is a full professor of agronomy at the University of Campania. Having previously held positions as a researcher and associate professor of plant physiology at the same university since 1999. She earned her degree and doctorate from the University of Naples, Federico II. Throughout her career, she has carried out research at prestigious institutions such as the Institute of Botany of the Ruprecht Karl University of Heidelberg, the Max Planck Institute for Molecular Plant Physiology in Gön, Potsdam, Germany, the INRA for Food Biology and Pathology in Bordeaux, Aquitaine, France. She has been a visiting professor at the Institute of Botany of the National University of Ireland in Galway. Her research has focused on the metabolic and physiological responses of agronomically important species and model plants to nutrient deficiency, saline stress, various cultivation methods, and the use of biostimulants, of course. Currently, she teaches agronomy, plant physiology, and post-harvest physiology. She is also a member of the scientific committee of biostimulant.com. So, this is Petronia's introduction, and I'm very happy to give the floor to Petronia for this presentation.
Hi, everybody. Today I will speak to you about how biostimulants can power up crop resilience, stimulating endogenous plants response that usually are activated only under stresses. Plants are continuously subjected to environmental fluctuation, that is physical and chemical changes that are physiological and non-pathological in the plant lifespan. However, when these changes became excessive or happen suddenly, they can be perceived by plants as stresses. In the last periods, the global changes, environmental changes, are affected plants with single or multiple stress phenomena. And this is a big issue because it is highly reducing the global production worldwide. Drought and high temperature are affecting all the world because of this rising of temperature. In the Horn of Africa, there is the worst drought in the last 30 years, and because since 3 years it's not raising. And in these regions, the water that is also called blue gold is an issue, a very important issue that can lead to wars and conflicts between nations and groups for the control of stable water supplies. However, also in the Mediterranean regions, prolonged drought and high temperature are causing critical issues for the water supply. And in the last years, the situation has became also worse because, for example, since January 2023 to September 2023, the temperature in Europe has risen of about 1.4 degrees over the average, and it has been the highest temperature recorded until the pre-industrial age. One of the reasons of this increase of temperature can be the number of people, because the European population has tripled in the last 70 years, and this has caused some consequences, such as the increase of the CO2 that has risen from 300 ppm from the pre-industrial age to 342 ppm in the 2023. Probably this is the highest CO2 concentration seen on the world since interglacial phenomena. And for this reason, Europe and Mediterranean basin has been defined a climate hotspot. The worst is that in the next years, it is predicted that the temperature can rise until 3.4 degrees before 2100, with a decrease of 30–40% of rainfall in winter season, and this is really a catastrophe. In fact, we can see that also in the Mediterranean regions, European regions, we can see a high to extremely high water scarcity. Clearly, the water scarcity is driven by demand, with the agriculture that accounts for 70% of water withdrawal. But as you can see, Italy, not only the south of Italy, but until the Emilia-Romagna regions, there is a very extremely high water stress. Of Emilia-Romagna is a demarcation line that has been signed in May 2023, when there was incredible floods that separated Italy into parts, creating a very arid zone in the south, with Sicily and Calabria that now are extremely arid for the 85–93%. High temperature and drought can affect plants by reducing photosynthesis. This effect is due to a double effect. The first effect is that the evapotranspiration determine a decrease of the water potential of soil and a rise of capillary rise of salts, determining a closure of stomata, which decrease photosynthesis. This reduction of photosynthesis rate determines an increase of ROS that affect globally plants, causing a decrease of plant growth and yield. In particular, high temperature and water stress can cause pleiotropic effects that, starting from the disturbance of photosynthetic electron transport, can cause ROS excess, oxidative and genotoxicity, membrane disorganization, disturbation to the water relation and metabolic processes, with the reduction of cell division and expansion, and for sure, problems to growth and development. Clearly, responding to stress comes with a cost, that is a cost that can be of 50–70 moles ATP for mole of osmolytes for obtaining osmoregulation and antioxidant response, and this cost for this cost, plants grow less because all the carbon and ATP are used for producing these protecting metabolites. Water stress, in fact, inhibits or delays seed germination by decreasing soil water potential, as told before, and in this way making plant uptake of water more difficult. This increases ROS and produce oxidative stress, and for the most, this release calcium or induce the release of calcium from apoplast and internal compartments, causing an APA, abscisic acid activation that delays dormancy. Also, high temperature inhibits seed germinations because the increase of temperature can, in a double way, accelerate initially the respiration rate, thus consuming very fast the seed reserves, but also at a certain point impairing this respiration. In fact, the germination percentage can decrease from 40 to 70% from 30 degrees centigrade, from the increase from 20 to 30 degrees centigrade. Drought can so directly increase APA synthesis and accumulation, and at the same time, high temperature inhibits the synthesis of gibberellins that have an antagonistic action with APA. And in this way, there is a prolongation of seed dormancy. Water stress, if it's mild, can increase the dimension and change the architecture of plants, for sure decreasing a little bit the dimension of plants at the beginning, for helping plants to uptake better the water and nutrients when there is a lower water potential. But if the stress becomes harder, the photosynthesis is impaired. Photosynthesis cannot more produce photosynthase to export to roots, and in this way, both roots and leaves stop growing, and plants' growth and survival is impaired. Moderate stress can cause thermomorphogenesis. In particular, heat stress in the first stages of plant growth, especially when temperature is lower than 37 degrees, that is a critical high temperature, determine an increase of hypocotyl and an elongation of root, causing thermonasty. In particular, hypocotyl grows for allowing leaves to move away from the soil that is very hot and to get more air and wind. This thermomorphogenesis is mediated by a phytochrome factor P4 that can induce auxin, and this auxin leaves the leaf blades going towards petioles and the hypocotyl, and in these tissues increase the production of brassinosteroids, and in this way increase the length of hypocotyl, determining thermomorphogenesis. Also, high temperature can affect endosperm and embryo development of the next generation. In particular, when there is a moderate high temperature that is lower than 37 degrees, This temperature can reduce grain filling and sites and decrease, for example, as in this peak, rice yield and quality. But if plants are subjected to severe temperature close to 40 degrees, this can cause grain malformation. Sudden and intense shock temperature can even cause plant death directly when the temperature is higher than 40 degrees, even if this high temperature lasts for a brief period. And this is the case of when there are the heat waves. High temperature can boost transpiration for allowing evaporative cooling even under drought. And this can happen for a dual phenomena. That is, the first, there is a physical phenomena in which water viscosity decrease, but also the cuticles became more permeable. But at physiological level, there is also an increase of K plus channels, potassium channels that became more permeable. Aquaporin increased the rate of water transportation, and there is also a change in membrane fluidity. Clearly, high temperature and water stress have an high effects on plants that is seen as a double stress. That is, it is seen as two main secondary stress from plants. That is, and perceived from plants in this way as oxidative stress and osmotic stress. This double stress alters structure and function of proteins, membrane, transport chains, and in this way impair cellular homeostasis. These changes in cellular homeostasis activate a series of signals, different signals, that is for the most calcium and ROS signals that activate stress gene response. It is interesting because chloroplast and mitochondria can communicate to plants or to nucleus and to entire plants that there is a stress. This happens because when there is an alteration of electron transport chain, this transport chain that doesn't work very well or isn't enough fast can create in any part of the complexes a rise of reactive oxygen species that can work as sensors and transducers of stress. In particular, also calcium, when there is a rise of temperature, because of the membrane became more fluid, there is an higher increase of calcium from the apoplastic compartment, in which the concentration can be very high, to the symplastic compartments, in which the calcium concentration can be 0.1 or 0.01 micromolar, and this influx of calcium can increase the calcium of 20 or 10-20 fold, and in this way, instead of activating ROS scavenging, when it works as retrograde signals, it activate ROS production like it happens before in the electron transport chains. And in this way, this enhancement of ROS is like a signals that activate expression of heat shock proteins and osmolytes for repairing oxidative damages and protective proteins. In this way, there is a response that can protect membrane and process integrity. Therefore, ROS and calcium waves that are generating during early shock or drought, especially as retrograde signals. For understanding what is a retrograde signals, we can do an example that is an retrograde signals is the one that goes from nucleus to chloroplast for governing the chloroplast or mitochondria function. For example, the small subunits of Rubisco are synthesized from nucleus and then transported to chloroplast for making Rubisco working. In this case, it is an anterograde signal, but there are signals that can go from the mitochondria and chloroplast, also from membrane, and make a signal for the nucleus, so create signals for the nucleus for activating transcription and cellular reprogramming. In this case, ROS and calcium waves function in this way, that is, their synthesis is works activating in nucleus or in other cellular tissues of the plants the synthesis of transcription factors for a cellular reprogramming in order to protect the structure and function under stress, but also for changing some physiological phenomena. For example, one of the response can be also stomatal closure. Therefore, exposure to high temperature that are not higher than 37 degrees or mild water stress are able to activate stress tolerance response, because can activate this transcription factor that enacts defense mechanism by means of osmolytes, metabolites, antioxidant enzymes, or also heat shock proteins that work detoxifying ROS, protecting membranes and proteins, and reestablishing cellular homeostasis and therefore stress tolerance. One of the important signals of the that calcium can operate is that of the overexpression of heat shock factors that regulate the expression of heat shock proteins. These heat shock proteins are very important, both for under high temperature stress, but also under other kinds of stress like drought, salinity, but also heavy metals. In fact, in this by this calcium signal, there is the activation of the ASFA1 factor that is responsible for the heat shock factors 70 and 19 that are very active in preventing the denaturation and aggregation of proteins. In this way, correcting protein misfolding. Moreover, HSFA1 can activate a second factor, that is HSFA2. Together, they form a superactivator complex that is able to increase the production of different heat shock proteins. You can see here 101, 90, 23.6, and heat shock factor 17.6. And in this case, in this way, they activate thermotolerance. Also, in high temperature and drought can activate the synthesis of osmolyte and secondary metabolites that work for protecting plants from oxidative stress and for in particular for stabilizing photosynthetic machinery and scavenging ROS. We have arrived to biostimulants. If we think about biostimulants. They are products that stimulate plant nutrition processes independently of the product's nutrient content, with the aim of improving one of these characteristics. One of these is tolerance to abiotic stress, and the way in which biostimulants enact this effect is because they contain molecules able of eliciting stress response and synthesis of osmolytes and antioxidants, but also they are able to elicit this ROS and calcium waves response. We start with showing the effect of algal extract biostimulants in priming techniques. These priming techniques can promote germination and tolerance to water, salt, and high temperature stress. These stresses are correlated also with salt stress is also related to water and temperature stress, because when there is evapotranspiration, salinity increases in the soils, and also when there is no rain that can lessiviate the soils. In this case, these priming techniques can increase the content of proline and glycine betaine, the enzyme activities of several antioxidant enzymes, and in this way protecting chlorophyll, carotenoids, increasing leaf area and the length of shoot and root of the plants, while decreasing the reactive oxygen species and the malonyldehyde, that is a marker of the lipid peroxidation. It is interesting that in a paper of Su of 2018, it was identified a motif in the promoter regions of choline monoxygenase and beta-enaldehyde dehydrogenase, that are genes involved in the synthesis of glycine betaine that are responsive to methyljasmonate. So methyljasmonate treated Citrullus lanatus cells stimulate the synthesis of glycine betaine even in absence of stress. Probably the induction of methyljasmonate glycine betaine cascade synthesis may be responsible for the effect of these algal extracts which contain methyljasmonate. Glycine betaine, that is induced by methyljasmonate in plants by the effect of because it is contained in algae. It's present usually in various halophytes and few cultivated plants, and for sure in algae. It is amphoteric, electrical neutral in a wide range of physiological pH, and is extremely soluble in water. Usually, osmolytes have these characteristics that they can be synthesized and accumulating in the cytosol without interfering with the cytosol function. And because they can work as osmoregulators, they are able to stabilize structure and activates macromolecules. It is interesting also that when there is dehydration, these osmolytes can substitute water in the water shell around the molecules. In this way, they preserve membrane and protein structure under stress condition, drought condition, or other stresses. And for the MOX, they work also as ROS scavengers. Glycine betaine in durum wheat under salt stress accumulates during prolonged stress only in young tissues, as you can see. And this accumulation is independent of nitrogen, but it has a special characteristics. That is, when glycine betaine is synthesized, it cannot be degraded. There are not enzymes for degrading glycine betaine. This means that when after stress, glycine betaine is diluted in other tissues. A second important thing is that plants synthesize glycine betaine only when it is strictly necessary. And in fact, in several studies, it is possible to see that in root apex of plants, in this case we see here durum wheat root apex and Arabidopsis root apex under water stress or under salinity. Usually the root apex of control cells has packed cells with very little very small intercellular spaces, but the root apex under stress change their structure. They show a vacuolization. They lose their typical organization, show plasmolysis, and lose the adhesions between cells. These damages can be caused by in this root apex when there is a delay in the synthesis and accumulation of glycine betaine. Quinn and Shabala in 2005 shows that the addition of 5 millimolar of glycine betaine to the culture medium can protect barley from osmotic stress. Again, also microbial biostimulants priming can enhance seed germination and tolerance to abiotic stresses. It seems that the phenomena of the protective phenomena are the same. Always the priming increase the content of osmolytes and reactive oxygen species like ascorbate and glutathione, but also antioxidant enzymes like superoxide dismutase or catalase or peroxidase. In this way, it protects important molecules like chlorophylls and photosynthesis, thus increasing leaf area and allowing also the export of photosynthase that make plants root growing. This allows plants to uptake more nutrients, like in this case phosphorus and potassium, and to have a water stress, an higher water stress tolerance. Also, plant extracts, protein hydrolysates, and directly metabolites can promote germination and tolerance to water stress by priming techniques. In this case, it is important to see that these biostimulants can increase DNA repair and cell division in the seeds. They make more nucleotides synthesized, and in this way, they increase shoot with vigor, increase germination, increase the relative water content while reducing reactive oxygen species and MDA. The priming with the same molecules can also increase the recovery from drought of plants. Always the system is the same. There is the synthesis and accumulation of osmolytes. Pigments and photosynthesis work better. The plants have an higher transpiration and stomatal conductance. There is an increase of yield and quality, an higher production of carbohydrates. At the same time, there is a decrease of hydrogen peroxide. In this case, the decrease of hydrogen peroxide is justified by the fact that there is a decrease of APA that cannot induce NADPH oxidase that is responsible for the production of hydrogen peroxide. It is also interesting that in this paper done on Brassica navus, under drought, the addition of glutamate can directly activate the enzymes involved in the synthesis of proline, like P5CS and P5CR, that are directly responsible of this increase of the content of proline. The accumulation of proline is one of the first signal of stress in plants. In fact, its accumulation is still debated because it is not clear if it's a symptom of stress, response to stress, or an adaptive strategies. Like glycine betaine, proline acts as osmolyte. It can stabilize membranes and proteins. It can detoxify ROS. It can control cellular redox potential. But it's interesting, you see here, that proline can also induce the gene expression in response to saline stress or to other stresses because there are genes that have proline responsive elements in their promoters and that can be activated when proline is synthesized after the first signs of stress. Proline, differently from glycine betaine, is rapidly synthesized in response to stress. Its synthesis depends on nitrogen content, and after the end of the stress, it can be degraded to provide energy, carbon and nitrogen for helping the growth after the stress. In this way, for this reason, is a spatial and temporal discrepancy in the synthesis of proline compared to glycine betaine. Osmolytes and exogenous secondary metabolites can induce also thermotolerance. We see here different metabolites like plasmonates. We have already seen that it can be responsible for the induction of glycine betaine. Arginine or putrescine salicylic acids, that is an activator of several response in plants, also of response related to the activation of mitochondrial electron transport chains and alternative oxidase. There is proline that can act also as biostimulants, ABA directly, again, putrescine, brassinosteroids, tocopherols, that is an important antioxidant, but also GABA. Of GABA is interesting. The role of GABA as osmolyte is interesting because it is not only interesting its accumulation, because it has been seen that it can cause also the accumulation of other low molecular weight osmolytes, but also because it works as scavengers and can protect directly photosynthetic pigments, photosynthesis, and photosystem to efficiency. It happens because it has multiple roles, but also its synthesis is particular, because in the synthesis of GABA from glutamate is the consumption of protons, and in this way its synthesis stabilizes pH. It has an antioxidant activity higher than the proline and glycine betaine. It can stabilize membranes and proteins. It can work as osmolyte. More important, its GABA shunt can be used for supplying anaplerotically intermediates to the Krebs cycle for making respiration restarting during stress, but also there is a strange thing that during the synthesis of GABA by means of decarboxylation of glutamate, there is a stoichiometric production of CO2 that can make photosynthesis, and in particular Calvin cycle restarting also when stomata are closed. Protein hydrolysate can reduce heat and rot stress. We see that, for example, in an experiment done on mice, protein hydrolysate at 12% of amino acids can induce, when they are used at high dilution, they can induce growth in early stages under heat stress, but also they promote leaf pigmentation, they increase proline, they increase antioxidant enzymes, they help increasing heat shock proteins and dehydrains that also help molecules to maintain their, and proteins to maintain their secondary and tertiary structure during dehydration. And they decrease reactive oxygen species and MDA. In an experiment done on tomato by Leporino in 2024, a protein hydrolysate from Malvace has been found to contain some peptides and omega-3 fatty acids that are responsible for the induction of stress response. That is, these dipeptides could be seen as inducers or as second messenger. And this is also interesting because this finding has been seen only in studies done on in food science. Again, algal extracts can increase drought tolerance directly on plants, increasing antioxidant metabolites, among which glutathione, ascorbate, and polyphenols, and also antioxidant enzyme. But again, compatible osmolytes like proline, glycine, betaine, but also soluble sugars. And in this way, they decrease ROS and MDA content, help plants to increase mineral uptake and increase also nitrogen use efficiency, and in particular, accumulating more minor amino acids and amides. These amides, and this can help plants to grow more, to accumulate more dry biomass and fruit yield. And for sure, this is due to an increase of photosynthesis and starch and sugars production. Ascophyllum nodosum extract, that is another algal extract, has been found to promote the tolerance to water stress because it is able to inhibit the expression of drought-responsive negative growth regulator at the shoot apical meristem. This can help plants grow, continue growing. Moreover, this Ascophyllum nodosum extracts, it is a commercial extract that has been used at 0.2% concentration by foliar spraying, can activate cell cycle, maintain active cell cycle progression by maintaining the expression of a histone H4 gene and enhancing the expression of a gene that promotes cell division at the meristem. This effect, together with the usual effect that we have seen promoted by osmolytes and antioxidant metabolites, helps plants to survive and overcome the water stress. only algal extracts can increase tolerance to drought and heat stress, but also microalgae itself used directly and as supplied directly to the roots. In this way, they can elicit thermal dissipation capacity, stomatal conductance. They can increase the number and of leaves and leaf area. They can increase the growth of root and stem, and in particular, chlorella is able to improve the production of secondary roots, or the growth of secondary roots, while Chlamydomonas can increase the concentration of micronutrients in roots and leaves. In these experiments, the microalgal extracts have been used together with their spent media, that is cultivation media, or they have used directly as cells. Like algae, some microbial biostimulants can increase drought tolerance. In this case, they enunciate ABA signaling cascade that, differently from the effect on seed, they can help overcoming stress by activated drought responsive genes and producing daedrins. In this way, they can help to increase the photosystem to efficiency, stomatal conductance, and plants growth. In this case, phytolactons can be used to change the shape of plants. That is, we can have plants that answer to drought, decreasing, or to thermal stress, decreasing their size, but continuing to function and to doing photosynthesis. There are other microbial biostimulants that are able to reduce the symptoms of drought and heat stress. In this case, there are the effects that we have already seen, but in this case, in addition, there is a decrease of ethylene and also a decrease of proline. This means that plants sense less stress. If the microbial biostimulants can inhibit the ethylene synthesis, plants sense less stress. The effect of these microbial biostimulants is due to ACC deaminases, that is an enzyme that inhibit or decrease the concentration of ACC, that is the precursor of ethylene, and in this way it makes plants sensing a lower stress, so not sensing stress. We can say that biostimulants can mimic the effect of pre-exposure to stress for inducing the acquired tolerance. In fact, if we expose plants to high temperature but less than 37 degrees or to a mild water stress, we can induce the plant response that is seen as the closure of stomata, accumulation of osmolytes, enhanced accumulation of abscisic acids that can help during growth or prolonged dormancy in seeds, can induce the expression of drought stress responses genes, but also activate the synthesis of activity or enhance the activity of antioxidant enzymes. If we use biostimulants, they can induce this endogenous response without being firstly affected by stress. In this case, biostimulants inhibit the secondary stresses, that is, make this osmotic stress and oxidative stress less dangerous for plants by stimulating retrograde signaling that is exerted by the release of ROS or calcium or MAP kinases. This make the upregulation, cause the upregulation of defense-related transcription factors or metabolites, or as we have seen also the peptides, increasing the ROS scavenging activity and osmoregulation, thus obtaining cellular redox homeostasis. In conclusion, if we understand better the modes and mechanisms through which plants respond to abiotic stresses, we will be able to develop biostimulants that can trigger endogenous responses to stress, even in absence of stress. In this case, inducing heat and drought tolerance. So, what we need is to continue doing both basic and applied research on cultivated plants for trying to understand the mechanism by which plants respond to stress, and at the same time, how plants' defense are boosted by biostimulants. I want to thank you for your attention, and this is also, and also the people who work with me. Thank you.
Thank you very much, Petronia. It was a very insightful presentation and very rich on this topic, which is nowadays essential. So now I will read some of the questions we received in the Q&A box. And if you have a question, I invite you to submit it now, because otherwise it will be too late. So first question. Do we have any insights into the role and mechanism of action of dipeptides in enhancing plant stress tolerance, particularly under conditions of drought and high temperature?
There is a paper, I have already made you see this paper here. I don't know. It's a paper of Leporino et al. From 2024. It is the only one that, okay, it's here. Sorry, I have this, it's okay here. Okay. They found in protein hydrolysate from Malvaceae that there were these dipeptides that they hypothesize that they can work as a carbon and nitrogen source when photosynthesis doesn't work, because there is for the stress, for drought stress, but also they can work as signals, and they reduce some stress compounds derived from glutathiones, and in this way they could decrease the content of ROS and mitigate the oxidative damage. This is very interesting, because in some other studies, medicine studies or food science studies, these dipeptides have been correlated to a different pathways. That is, for example, pyroglutamyl peptide, because one of the dipeptides that were found was pyroglutamyl dipeptide, can be produced by lactobacillus for inhibiting the expression of virulence genes for toxic shock syndrome. And this is interesting, because this means that these dipeptides can have a control on gene transcription or gene expression. And also they can work as signals. So they are an open field to do research, because also they are found in many foods, for example, in soy sauce and other food, in Japanese food. They are correlated to the umami test, but also to a strong antioxidant activity. And they have also been correlated to some benefits for health, like hepatoprotection, a direct effect on hepatoprotection, or they have been found as antidepressive or anti-inflammatory. So they are interesting to study because a lot of different metabolites that have been found working in plants, in animals as antioxidant, or that have an antioxidant effect or carcinogenic effect in cells, in animal cells, have been found also having important effects in plant cells. I mean, some of the minor amino acids, also GABA, have these important effects, like the peptides.
Okay, great. Thank you very much. So another question. We have 10. What are the osmolytes molecule analysis you recommend in order to compare study plans regarding abiotic stress tolerance?
Okay, so the first one for sure, proline, but also an increase of soluble sugars. It can be seen as an important osmolyte effect, so an effect for osmoregulation. It depends if there are POASE, that is cereals, glycine beta is also important. For the most, I have seen that the targeted analysis of amino acids can help me to understand if there is an effect, a stress effect, and how plants respond. Because the first response of plants is not a secondary response, because the secondary metabolism response is more expensive. For the most plants, answer to stress with the primary metabolites, for example, proline, that is an amino acids, but also sugars is important. Also, they change the amount of the amount and profile of minor amino acids. So for the most, the easiest are proline and soluble sugars. These are the first, for sure, the first to check. And they can also check the amount of total polyphenols, because these act as important antioxidant, and they can also protect photosynthesis during any kind of stress.
Okay, thank you very much. Does an increase of secondary metabolites such as flavonols or anthocyanins be a sign that a plant is more stressed, or it is a sign that the plant is more tolerant against drought or hydric stress?
It depends on plant and also on the specific response. Sometimes plants, also different ecotypes of plants can answer differently to stress. We made a study on the metabolic profile of 7 ecotypes of Pomodorino, del Piennolo, del Vesuvio, that are the little small tomatoes that are produced on the volcano on the Vesuvio. And the profile of these ecotypes changed a lot, showing that some of them used lycopene, for example, more lycopene, or other anthocyanins or polyphenols, other used amino acids or sugars. So it depends on the ecotypes and the specific response. So we cannot tell if we do not study and we don't see the effect in compared to not stress control.
Okay, so next question, we'll take 3 or 4 more. So thanks for the great presentation. And we have a couple of messages here that are congratulations for the presentation. Can you clarify which are the differences in terms of bioactive compounds between algal extracts and protein hydrolysates? Can we expect different effects on plants?
Algal extracts are really complex. We tried in 2017 to make a complete analysis, so we wanted to see by untargeted metabolomics, but trying also to understand what was the real amount of these compounds into commercial algal extract. And the first was that for the first months, they were unable to extract and to see these compounds by GCMS or LCMS, and the lab was that of Max Planck Institute, and the group was that of Fermi, that is one of the main researcher working with metabolic profiling by plant spectrometry. This because the profile is complex, very complex. There are all the possible classes of metabolites. Among these, polyphenols, proline, glycine, betaine, different sugars, carrageenan, alginate, mannitol, everything. There is everything, and also amino acids for sure. While protein hydrolysate has for the most amino acids and peptides, because there is a fractionation of the content of proteins, of the protein content. And so it's a more defined compounds that is we can really characterize, and it has been done. For example, Colla has characterized the fractionation for the dimension of peptides and the aminogram. So we see that effect is really due to the amino acids or to the peptides, while with algae, the effects are multiple. We have seen also that they contain jasmonate, and that this jasmonate can induce a huge amount, but also salicylic acids. Studying algae is more difficult than studying protein hydrolysates because the composition is much more complex.
Okay, thank you very much. Another one, how does GABA contribute to the reactivation of photosynthesis under conditions of stress, such as drought or high temperature?
We have I have a slide here. This is the GABA one. Okay. GABA has multiple roles. For the first, like proline is synthesized starting from glutamate, that is one of the two first amino acids produced in the nitrogen organication. So the glutamate is always present, and GABA can be produced always. Especially GABA, it's produced when there is a change on pH. So when there is a cellular pH, a change of the pH status, the glutamate decarboxylase, that is the enzyme responsible for GABA synthesis, is activated, and in this way, because it consumes protons, stabilizes pH. Once GABA is synthesized, the GABA shunt can reactivate or can produce again glutamate, because GABA is used as an aplerotic compound for producing succinate that is fed to carboxylic acid, making again alpha-ketoglutarate for the production of glutamate. Until here, this was what was known until a few years ago. But then it has been discovered that GABA has this very high antioxidant potential, higher than proline and glycine beta. Moreover, during its synthesis, stoichiometric amounts of CO2 are produced, and these are able to make photosynthesis restarting. And in particular, it was seen in some papers done on durum wheat under salinity, that under high light, so high light, drought, and salinity, when the plants were grown under high light, that inhibited the production of glycine beta and induced the death of GABA, the plant had better photosynthesis than under low light. This means that GABA, synthesizing the synthesis of GABA that increases the concentration of CO2, helps the photosynthesis to restart and makes plants being better, even if there are at that moment multiple stressing instead of a single salinity stress. I hope to have been clear. If not, I am open to explain again.
Okay, I think that now it will not be possible because we are running out of time, but of course they can leave the question on the biostimulant.com website and you'll be able to answer later. So the last two questions. What effects does the moderate temperature, for example, thermomorphogenesis, have on cell proliferation? Or is it just the elongation growth that is stimulated during thermomorphogenesis? And I think the same as two different questions. And does shoot and root meristems behave differently to the moderate temperature?
We have here thermonosty. Okay. When there is auxin, when there is auxin and brastinosteroids, usually the effect is cell distension. So it's not on cell division, but it's on an increase of cell size. So the cells elongate instead of divide. With the same effects exerted by photomorphogenesis. The photomorphogenesis induces an equal distribution of auxin, and in this part of plants in which there is this unequal distribution, there is auxin activate ATPase, and so the acidic growth of cell, so a distension of cell. The same effect can be seen when auxin and brastinosteroids work together for inducing thermomorphogenesis. This is also the same effect, even if in different plants, of the escape from shape. That is, all these effects are exerted by auxin by inducing an elongation of cells.
Thank you very much, Petrania. So the last question, can you clarify how retrograde signaling is activated under the two stress conditions?
Okay, it's here, somewhere here. I will find it. Okay. Okay. We have two kind of retrograde signals. The first one is when there is an increase of temperature or when stomata closure, we have a dysfunction of membranes. The first is when stomata closure, there is a stomata closure because there is drought. So plants sense that there is less water. That is, the water potential of soil is lower, and so plants have problems to uptake water. And they, in this case, directly activate APA for the closure of stomata. When stomata closure, there is a decrease of CO2 in the mesophyll cells, and so the Calvin cycle decrease its activity, not consuming NADPH and ATP that are produced during photosynthesis. When NADPH is not reoxidated, the photosynthesis lacks its final acceptor. For this reason, there is a decrease of electron transport rate, and in any case in which there is a decrease of this or the efficiency of this transport, you see in any part of the complexes there is the production of reactive oxygen species. And this is the first retrograde signals. That is, and this works also for mitochondria, because when there is an increase of temperature, the membrane became more fluid, and the electron transport in the mitochondria is less efficient because the complexes starts moving from their position. That is the best position for increasing the efficiency and the rate of electron transport. And this is the retrograde signals from electron transport chains, that is from mitochondria and from chloroplasts. But there is also another signal, that is that of calcium, because that of calcium also derived from the fact that ROS alter the profile and structure of membranes, decreasing or increasing the polarization of the membranes, or the membranes became more fluid because there is an increase of temperature and the channels open, leaving calcium entering and doing its job as second messenger, activated the retrograde response.
Thank you very much. And this was our last question for today. Thank you very much, Petrania, for this presentation and for giving us some time. And I would also like to thank everybody for attending the webinar today. I repeat, if you still have questions, submit them directly on the biostimulant.com website in the specific section, Ask the Expert. I would like to remind you that the recording will be available soon on biostimulant.com website and on our social media. And I have a nice day, and I hope to see you again very soon in one of our webinars. Have a nice day, everybody. Bye.